this remarkable set of images from Japan, I am hopeful saw this image of this woman looking for her son in the rubble of the quake and the tsunami. And of course, it's wonderful to know that a year later she found her son, he's fine, there he is, there they are, in the exact same spot. Now, if you don't get B-Space emails, check your email settings, because the emails that I send sometimes contain links like this, but most of the time contains things that have to do with helping you do well in the class. So make sure your settings allow you to do this. Now, at the end of the day, everything is relative. There are, quote, first world problems, and then there are third world problems. There is a long line for your morning latte, and then there's death of schistosomiasis at age 5. And then there's everything in between. But I think as far as sort of gradients of pain, one would imagine that what this woman was going through at this moment is at the very, 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 very bottom. This is as awful as it probably gets. Now, I don't suppose by chance some of you have the misfortune of knowing, I'm hopeful not from direct experience, in medicine, what is the worst kind of pain, objectively speaking? So what is the worst kind of pain that a human being can experience? No, not mental. Anybody know? Not birth, nope. It's cancer pain, metastasis. The metastasis of tumor. Cancer pain is so awful that most patients plead with their physicians to end the suffering. The current standard of care for cancer pain is a lozenge called Actique. It's essentially an incredibly powerful analgesic. I think it's a derivative of fentanyl. And what you do is you give the patient the lozenge and they suck on it for a little bit until the pain goes away. But it also depresses the breathing center, so they start to choke. Then you take the lozenge out, and they sort of die like this. The reason I'm telling you this early in the morning, is to tell you a story about a boy in Pakistan who unfortunately died. But his death and his family provide an invaluable service to the biomedical community. This was published in Nature, and I'll show you the reference in a second. He was apparently born with a congenital inability to sense pain. So he was 10 when he presented himself to the medical service. He performed street theater. So he would place knives through his arms and walked on burning coals, but experienced no pain. This kid died before being seen on his 14th birthday after jumping off a house roof. So scientists, while of course mourning this tragedy, were able to work with members of his family and his own genetic material to identify the biological basis of this remarkable feature that this boy had. And the biological basis was a mutation in a gene called SCN9A. It encodes a protein that is expressed in your spine. And in fact, the spine is the central organ of your body that transmits pain. So his inability to sense pain had to do with the inability of his spine to transmit the pain sensation to his cerebral cortex. But importantly, the boy was otherwise just fine. There was any pathophysiology associated with that mutation. This is tremendously exciting because this means that if biologists and physicians and chemists find a way to inhibit the activity of this protein in conventional human beings, that small molecule, that agent that inhibits that protein, will be the best analgesic on Earth. And there are many drug companies, both large and small, currently working on finding such molecules because they are convinced that such an inhibitor would be a much better agent for the alleviation of the tragedy of cancer pain than a lozenge with a breathing center depressant. Was his DNA sequenced in its entirety? No, it was not. What ended up happening is they studied a number of families in which this remarkable trait appeared, and they identified the gene mutations in which cause this remarkable remarkable human trait, using mapping by linkage. And by now, I'm sure you appreciate why did I start this morning's lecture with this, because we're going to discuss briefly how genetic mapping works. But we're not going to discuss this using humans. We're going to discuss this using fruit flies. The first such maps were constructed by an undergraduate in Columbia, Alfred Sturtevant, and the foundational principles according to which they mapped genes back then have been preserved without any change. So today, a century later, geneticists more or less map by linkage exactly the same way. And in order to walk you through how one maps by linkage, we need to discuss briefly this very famous experiment. It's very famous dihybrid cross that Alfred Sturtevant, an undergraduate in Thomas Hunt Morgan's lab at Columbia, did. You will recall that dihybrid crosses is when you take two pure lines, and pure lines is not a term from calligraphy where, oh, the line is so pure. No, it's an animal when crossed to itself, or a plant that when crossed to itself, or another plant just like this, produces progeny just like it. So in genetic terms, of course, a pure line is a group of organisms homozygous for all alleles of loci that control the trait of interest. Incidentally, students of ancient history, has there ever been a pure line in humanity? Anybody know? No, more or less for much of the genome. You're saying no. Actually, think about this. Ancient history, think royal families, Egyptian pharaohs, Nefertiti, this model of ancient beauty, was the offspring of 7 consecutive generations of brother-sister marriages. That's more or less how you make pure lines. You brother-sister cross. Nefertiti was a pure line. But these days, the two traits under study are eye color and wing shape. It's useful to work with traits that one can easily score. You simply look at a fly and ask, listen, do you have normal wings or do you have these very cutesy vestigial wings? Do you have conventional red eyes or do you have purple eyes? In this cross, homozygous dominant animal is crossed to a double mutant animal. All the offspring are, of course, heterozygous genetically, but phenotypically they all look like mom. If you then take these F1 hybrids, and rather than crossing them to each other, as Mendel would have done, let's test cross them. Please write this down. I want you to come to the midterm knowing the meaning of the word test cross. It's not a cross you do again after your first cross to confirm that you didn't mess up on the first cross. In genetics, test cross means something very special. Specifically, it is taking an animal of interest and crossing it to another animal that is homozygous recessive for all alleles of all loci for the trait under study. The reason it's called a test cross is it allows us to immediately reveal that does the animal being tested, despite looking exactly like homozygous dominant grandma, is that animal homozygous or heterozygous for the loci? Small homework. How would the results of a test cross differ if you take an animal homozygous dominant for all the traits, for all the loci, or an animal that's heterozygous? They are phenotypically indistinguishable. When you test cross them, however, they will look different, the offspring of the cross. In what way? Look in your textbook. Ask your GSI. Don't Google this. I have nothing against Sergey and Larry. Don't Google this. Ask somebody who knows, somebody with a pulse at that moment. In stunning contradiction to Mendel's second law. Let's do this in real time, the way that people used to do this with blackboards. if you take an animal that looks like this and test cross it, you should get progeny in all four classes, big A, big B, big A, small a, big B, small b, at a ratio of 1 to 1 to 1 to 1. Now, some of you in the 8:21 a.m. morning days will be saying to me, we thought it should be 9 to 3 to 3 to 1. Yes, it should be 9 to 3 to 3 to 1 if you do a cross like this. But if you do a cross like this, also known as Control Z, then you should get a 1 to 1 to 1 ratio. Tiny homework. Why, according to Mendel's second law, should the offspring of this cross yield progeny at a 1 to 1 to 1 to 1 ratio? Before you say, together with Calvin and Hobbes, why do we care, is the real question. The answer is, you can't really be a physician or a scientist or a public health professional without knowing why the answer to that question is what it is. It's fundamental principles of modern genetics. So what one should get when crossing doubly heterozygous purple vestigial animals, you should get all four types of offspring in the following ratios. Instead, I'm very sorry to say, poor Alfred Sturtevant got a massive representation of the the grand parental classes. The expected ratio from counting 2,839 flies, and before you fall asleep, as you fall asleep, count them. 1 fly, 2 fly, 3 fly, 4 fly, and so on. And don't forget, you're not just counting them, you're phenotyping them. So you're looking at each one and you're scoring which kind of wings it has and what kind of eyes it has. If this kind of work appeals to you, join us. There's a lab on campus currently doing this. I highly recommend it. It's certainly better than watching stock prices move up and down. It's a lot more gratifying. So, a massive overrepresentation of things that look like grandma and things that look like grandpa. In fact, as a student pointed out to me at the end of last class, there is a typo in the slideshow. That's what this should look like. You're expecting ratios that look like this, and the outcomes are things that look like that. This is in such violation of Mendel's second law that initially these data induced some sort of, well, paralysis is perhaps too strong a word, caused a slight feeling of being overwhelmed. Mendel's second law and these beautiful ratios, 1 to 1 to 1 to 1, only work for loci that are not genetically linked. What does genetically linked mean? It means lie on the same chromosome, less than 50 map units away from each other, or 50 centimorgans. Same chromosome, less than 50 map units away from each other. Let me explain to you what both of those terms mean. Sturtevant realized that the extraordinary overrepresentation in the second generation of this cross of animals that look like grandma and animals that look like grandpa has to do with the fact that alleles of genes that affect the trait differences in this cross, so the alleles of the gene that affect eye color in this cross, purple, and alleles of the gene that affect wing shape in this cross, vestigial, lie on the same chromosome. And for Mendel's second law to play itself out, let's say round versus wrinkled and yellow versus green, they have to be on different chromosomes, such that the homologs can do their little, I'm not talking to you, you're 2, I'm 1. And 2 is like, whatever. Such that when this animal in a test cross makes gametes, the majority of the gametes will retain the alleles of purple and vestigial in their original configuration, the way they arrived from the grandparents. And as I look at you, I see, 20% going, 20% going, and about 60% going. Good. This is actually relatively straightforward. So what I need you to do, now that you've listened to me walk you through this once, is please study this. You have the long weekend ahead. Well, not long. You have Friday, Saturday, Sunday, and you'll say to me, but we have all these other classes. Of course you do. But my goal here is to tell you what I'm going to ask you about. I will ask you why, in this experiment, after all of this dynamics of crosses, this to that produces that, that test cross produces this very bizarre skewing. What explains that skewing? The answer, they're on the same chromosome. Why does the fact that they're on the same chromosome explain this remarkable skewing of ratios? I promise you this is straightforward. I promise to ask you about this on the midterm. Will I ask you about this kid in Pakistan who was congenitally insensitive to pain? No. Life will ask you about that. But on the midterm, I will ask you about the technique that allowed the scientists to map the gene, the mutation in which gave that boy that appearance. Okay. Since they're on the same chromosome, they should never obey Mendel's second law. Alleles of genes inherited from grandparents should stay together forever, like in that awful Rick Astley song. We are thus stuck with explaining where this relatively unabundant group of animals come from. They appear to have, and here comes a really neat word, they appear to have phenotypes that are recombinant. What does that mean? That means mix and matched, shuffled, relative to what the two parents provided them with. Enter the California slender salamander round 2. I don't know what it was with cytologists in the late 19th, early 20th century. Did they all travel to Berkeley to catch these and work with them? But remarkably, not only did this lovely little critter provide the world with mitosis and the word chromosomes, it also provided the world with dynamics of meiosis. A different biologist in the late 19th, early 20th century looked at spermatogenesis in Batrachoceps attenuatus and observed these remarkable shapes. That scientist hypothesized that what one is witnessing is the exchange of bits of chromosomes. Sturtevant and Morgan correctly realized that these rare recombinant phenotypes, so an arrangement of traits not seen in the parents, is due to a recombination of the genetic material. Specifically, that chromosomes do this. This is actually not a figure from your book, but I couldn't find a figure this clear in your book. So I used this figure from an upper division class I teach, which is upper division genetics. Here are the parental chromosomes. Here's the purple vestigial homozygous wild type from wild type mom. Here's the lowercase homozygous recessive purple vestigial from homozygous recessive dad. Here's meiosis in their offspring. And that, folks, is a crossing over event, which produces recombinant chromosomes. Now, if you take upper division genetics, I will teach you that these types of things have been actually mapped in humans. In a stunning act of the power of modern genetics, scientists have taken parent-child trios, so both parents and the child, to map comprehensively across the entire genome of a living human being where in his or her parents crossing over occurred on all 22 autosomes. We have a high-resolution map. And I appreciate the fact that not all of you think of that and go, ah. Some of you go, oh. But, oh my goodness gracious me. How much time do we have? 29 minutes. Okay. This young lady has asked, why would we want to map crossing over events? The human genome, don't write this down, this will not be in the midterm, I'll talk very, very fast. The human genome contains multiple genetic signatures of risk. Cardiovascular disease, diseases of the central nervous system, Alzheimer's, schizophrenia, Parkinson's, susceptibility to cancer. Unfortunately, the human genome is partitioned into large chunks that appear to travel together through evolution. So it is not the case that recombination can occur between any two genes in the human genome. The human genome is partitioned into large, I guess, domains called haplotypes. Don't write this down. This will not be on the midterm. This will be on life. Thus, well, it depends on what you want to do. If your goal is the life sciences, then believe me, your life will center around haplotypes. This means that the human genome travels through generations as blocks. So understanding where those blocks are helps us understand where the genetic signatures of disease are, and knowing that helps us understand what caused disease. Knowing that, in turn, helps us either prevent it or treat it. And I realize I've sort of not answered your question. I've just thrown a bunch of terminology at you. Two upper division classes at Cal, 104 and 140, and this is taught in both of them.
So it shows, so we would know which chunks travel together.
Correct. This finding that the chromosome, to you, this is sort of middle school material. Seventh graders probably learn this. There's a chromosome, it's a piece of DNA, it contains alleles of genes. Next, boring. Let me show you, folks, what the model of the chromosome was at the time. This was 1912. This was from a professor named William Castle, who was otherwise quite brilliant. He founded mouse genetics. And his model of the chromosome was this: row, row, row your boat. Let me emphasize, this was his idea of how a chromosome looks. It was this three-dimensional shebangy, for lack of a more accurate scientific term. What was this based on? It was based on his conviction that an organic molecule, such as DNA, could not have a simple linear form. I'm not joking. He was really against the linear model of the chromosome because he didn't think that DNA could be long and linear. Unfortunately, this was 40 years before DNA was shown to be long and linear. This is from Sturtevant's own memoirs. Now, I want to emphasize that he's describing a time when he was a senior in Morgan's lab. Castle published a diagram to show the interrelation of genes affecting the colors of rabbits. They showed the spatial relationship of genes in the nucleus. In the latter part of 1911, in conversation with Morgan, his mentor, about this attempt, I love the next turn of phrase. In our modern times, which are laden with YouTube comments that cannot be shown to small children, we mostly tend to call each other names that cannot be printed in polite society. Back then, people used to say things that were just as insulting, but in much more understated ways. In conversation with Morgan about Castle's hypothesis, which we agreed had nothing in its favor. Ah, sir, your hypothesis has nothing in its favor. I suddenly realized, let me emphasize, this is like a 20-year-old, quote, kid, unquote, who changed the course of genetics. I realized that the variations in the strength of linkage, already attributed by Morgan to differences in the spatial separation of genes, offered the possibility of determining sequences in the linear dimension of a chromosome. I went home and spent most of the night, to the neglect of my undergraduate homework, in producing the first chromosome map, which include the sex-linked genes yellow, white, vermilion, I don't know what M stands for, and R for red, in the order and approximately the relative spacing that they still appear on the standard maps. The frequency with which a recombinant progeny appear is directly related to the distance between the cognate genes on the chromosome. The unit of genetic distance is named after Thomas Hunt Morgan. It is 1 Morgan. 1% recombination is 1 centimorgan. A very simple number for you to remember. What is the relationship between genetic distance, so the likelihood that 2 genes will recombine, and how far they physically are apart on the chromosome? On average, 1 centimorgan of genetic distance in the human genome is 1 million base pairs. So 1 centimorgan is 1 million base pairs. Before I transition to the next part, I want to highlight the remarkable power of genetic analysis. Today, when one looks at genes, what you do is you actually sequence the DNA. So you take the DNA and you put it in a machine, and 3 weeks later, after having spent maybe $5,000 to $20,000, you have the complete sequence of the genome. All the facts that I've presented to you so far in this section of the class, that there are genes that lie on chromosomes, and that there are differences in genes, explain differences in traits, that alleles of genes behave a certain way during meiosis and are arranged in a linear order on the chromosome and can exchange bits via recombination. All of this was complete by 1930, 23 years prior to the discovery of the structure of DNA. Purely by crossing animals or other types of creatures. Genetics is that powerful to this day because it allows us to indirectly observe the workings of the living organism. Now, there are other, of course, remarkable fields in which indirect observation is very powerful. Does anybody care to name for me a very large experiment in the field of physics, which is currently doing an indirect detection? It's in, I believe, in Switzerland and, sir? The speed of neutrino is one. The Opera experiment, apparently the neutrino is slower than they claimed 6 months ago. I was thinking of an even larger one. It's this large ring. Sir? That's the search for the Higgs boson. The Large Hadron Collider, the LHC. Google it. It's remarkable. Just visualize what the LHC looks like. It's, I think it's a circle. The diameter is larger than the Berkeley campus. It's a very large object. And it aims to find the Higgs boson. You cannot see the Higgs boson. You have to detect it indirectly. Mendel didn't sequence DNA. He didn't even know DNA existed, but he divined the fact that there are genes and alleles of them. Similarly, Sturtevant did not sequence DNA, but he divined correctly that alleles of genes that affect the 2 traits under study lie in a linear order on a chromosome. That power of indirect observation and inference is partly what attracts people to genetics, because it's so pure in its slight separation from the way the thing works at the molecular level. It doesn't matter. We still figure out how life works. So, by the way, part of the reason I put this together is to provide you a small study guide for the midterm. Each one of these claims has an experimental foundation that we discussed in this section of the class. So as you're preparing to study for the midterm, look at this slide and ask yourself, what is the evidence that genes lie on chromosomes? What specific experiment did we discuss that that's the case? What specific evidence was provided in that experiment supporting that conclusion? And so on, and so on, and so on. No. I mentioned to you that an important challenge for your generation, and my generation, of course, as well, is try to understand the connection between human genetics and human traits. I also mentioned to you that the relationship is not a simple one. Think back to human height, which appears to be controlled by hundreds of genes. But one has to start small. You cannot swing for the largest problem straight out because you'll just hurt yourself. So I'm going to walk you through some remarkable experiments that started to connect genetics to the chemistry of life in a way that has completely revolutionized the way we think about biology and the way that really is the way forward that the 21st century will use to provide answers to why heart disease run in families. And I'm going to show you two examples before showing you an experiment. I don't know if I will have time to do all of this right now. If not, fortunately, there's still Monday. But the opening two examples, I am very hopeful you will remember for the rest of your life. There are two specific short things from them I need you to know for the midterm, and I've summarized them at the end of the sequence. For now, folks, I give you a picture. There is something uniquely human about this. Only humans do something that you see before you. Now, it's not advertising. Now, of course, only humans do advertising. And it's not the fact that humanity appears to have created a situation in which a 10-year-old knows how to work the camera as if she were ready for the runway in Milan. There's something uniquely human about this. Anybody care to tell me? Very good. The consumption of milk as an adult, with the possible exception of domestic cats. What mammal drinks milk as an adult? Where would you get milk? Imagine you're a wolf, an adult wolf, and you want some milk. Are you going to milk the neighborhood cow? Are you going to go back in the den and say, "Hi, Mom, remember me?" What would Mom say to you? As this article tells us, in most humans, the ability to digest a lactose, the main carbohydrate present in milk, you don't need to know this chemical structure. You will need to know it if you take upper division biochemistry. So, for example, MCB 102. For now, this is just a disaccharide. The main carbohydrate, the ability to digest lactose, the main carbohydrate present in milk, declines rapidly after weaning because of decreasing levels of the enzyme lactase. Lactase is predominantly expressed in the small intestine. What does that mean, lactase is expressed? This means that there is a gene in the human genome called lactase, and it is on in the small intestine. It's not on in the muscle or the skin, because we don't eat with our skin. It's on in the small intestine, because that's where the milk goes after you drink it. It is expressed in the small intestine, where it hydrolyzes lactose into glucose and galactose. Glucose and galactose, sugars that are easily absorbed into the bloodstream. So in most humans, the ability to digest lactose declines rapidly after weaning. However, some individuals have the lactase persistent trait. And remarkably, the frequency of lactase persistence varies dramatically from population to population. For example, it is very high in Northern Europeans. Over 90% of Swedes and Danes have the ability to digest milk as adults. It decreases in frequency across Southern Europe and the Middle East, 50% in Spanish, French, and pastoralist Arab populations, and is low in non-pastoralist Asian and African populations. So, for example, 1% in mainland Chinese, 5 to 20% in West African agriculturalists. Of note, however, lactase persistence is common in pastoralist populations from Africa, so 90% in Tutsi and 50% in Fulani. Interestingly, and I like this sentence because it reminds us of the difference between genotype and phenotype, lactase persistence is inherited as a dominant Mendelian trait in Europeans. Small bit of homework. What does that sentence mean? Is inherited as a dominant Mendelian trait. Please make yourself a note to ask your GSI. Look this up in the textbook, or ask me during office hours, such as the ones that are coming up right after this lecture. Come to the midterm being able to answer the question, what does the sentence mean? Trait X is inherited as a dominant Mendelian trait. Okay, that's how it is inherited. So we look at pedigrees, we look at whoever this is. Taylor Swift, right? Okay, we'll look at Taylor Swift, and we look at her family, and we observe the inheritance of this trait. But this is a biology class. What underlies that difference? Well, what provided a remarkable clue is the fact that it is a recently evolved trait. Namely, the ability to digest milk as adults arose in human beings coincident with the domestication of cattle, but only in those cultures that practice dairy farming. And incidentally, I'm showing you an image of Usher, who's obviously African-American, and Anna Ivanovich, who's obviously Caucasian, because I'm about to tell you that while they both have this trait, adult lactase persistence, it appears to have evolved independently in them. Yet the genetic signature converged on the same position in the genome. To show you that, I need to show you a picture of Berkeley's pride and joy, Aaron Rodgers. Go Bears. And in a wonderful pedagogically appropriate example, here's a little African-American kid wanting Aaron Rodgers' milk. My point to you is, while they both can digest it, that didn't come out quite right, his glass of milk. You know, very briefly, since I only have 14 minutes left, in Mother Russia I learned this proverb: people perceive the world according to the level of their internal corruptedness. Каждый все понимает в меру своей испорченности. Seriously, you know what I was going to say. Come on. So this is a marvelous example of convergent evolution. Shh, please. Of convergent evolution and natural selection in action. The allele in Aaron Rodgers and this kid are not different forms of the enzyme. They are different forms of the regulatory region of the gene, which happens to sit in an adjacent gene. Remarkably, while evolution converged on the same position in the genome, it yielded different alleles that have the same effect. Let me walk you. So this is the primary data, which I understand is largely unintelligible to you. I made this little PowerPoint animation to explain to you what's going on. The lactase gene has a regulatory element that keeps it on. It's called an enhancer. It's on the same chromosome. It's just not inside the body of the gene. The function of this enhancer is to keep the gene on in children, and then right after weaning, the enhancer shuts down and the gene goes off, and lactase is no longer made, and voila. Under selection from the domestication of cattle, in Europeans, and in fact as recently shown in Indians, the majority of Indians, an allele evolved that allows that enhancer to stay active longer into adulthood, and thus the gene is expressed longer. Remarkably, experiments have shown that in East Africa, which practices dairy farming, the same gene also evolved to allow persistent enhancer function, but it's a different genetic form of the enhancer. I'll show that in a second. Most astonishingly, this was literally last year, they found the Saudi Arabian allele, which affects the same enhancer, which also causes persistence through adulthood. And it's a different form of the enhancer yet again. What does that mean, different form? It means when you actually sequence the DNA of those four groups of people, Europeans and Indians, East Africans, or the Saudis, you observe mutations in the same stretch of DNA, but they are different mutations. Different mutations causing the same biological outcome. I don't suppose any of you remember an example from last class in which we discussed different mutations that cause the same biological outcome. Silent mutations was one, had to do with a human genetic disease. Cystic fibrosis, thank you. Many different mutations in the CFTR gene can cause cystic fibrosis. And I talked to you about the fact that this means that the gene is a unit of biological function. I put this story in here by way of a preview that how we define gene depends on who we are as biologists. Here I am showing you that a gene in humans have evolved to adapt to dairy farming, but the mutations that are the signature of this adaptation are not in the part of the gene that encodes the protein. It's in a regulatory element. What I'm about to show you is borderline science fiction, except it's completely real. They found human remains from the Neolithic, so right around the time when cattle was being domesticated, and they genotyped the remains at this locus to ask, did those ancient Europeans, were they already lactase persistent or not? And the answer is, oh, by the way, I forgot to mention, talk about convergent evolution. So the Europeans, the Indians, and the East Africans evolved lactase persistence because they domesticated the cow. The Saudis drink camel milk, which is very nutritious and I'm told quite tasty. I've actually never had it. So here's an example of two distinct kinds of selective pressures which act on the same biological circuit to produce a genetic signature of adaptation in the same position in the human genome. So, my goodness, talk about evidence of human history. We're used to thinking of fossils or pottery or other remains. This is genomics. So these individuals identified 8 Neolithic skeletons from Central, Northeast, and Southern Europe, ranging in age from 5800 to 5000 BC. They genotyped, they sequenced. Can you, somebody hypothesize what technique they used to sequence? How did they pull out the DNA? PCR, of course. They used PCR. They PCR'd out that ancient DNA from these skeletons, and they did not observe the allele most commonly associated with lactase persistence, meaning that before the onset of cattle domestication, lactase persistence was rare. And then enter the cow. And then a different study looked at late Neolithic burials from the Basque country from 5000 to 4500 BC, and they found an increase all the way to 27% of the alleles of the population, now encoding lactase persistence. But most importantly, this frequency is much lower than it is today, fully consistent with the idea, with the remarkable idea, that it was in fact the cow or the camel that caused humans to genetically change, only in some parts of the world, but not others. I find it astonishing, I must admit to you, that scientists have now the techniques to trace, once upon a time, the cow was domesticated. Who alive was there? Or who alive then is around today? Well, no one. But the DNA is still there, and our biology to parse biological circuits is, et voila, a little trace of human history. I need to show you, I have 8 minutes left, and in which I'm going to teach you another example which is just like this, and with that I'm going to let you go, hold office hours, and then continue Monday. Nicholas Wade, who writes very nice articles in the New York Times about all sorts of things biological, I really encourage you to read his articles. They typically appear on Tuesdays. Tuesdays is when the New York Times has a science section. Has written an article about an absolutely stunning finding that came out 2 years ago, which explains why the inability to tolerate meaningful doses of alcohol is highly prevalent in some parts of the world and not others. You're not going to believe what explains it. 10,000 years ago, people in southern China began to cultivate rice and quickly made an all too tempting discovery. The cereal could be fermented into alcoholic liquors. Carousing and drunkenness must have started to pose a serious threat to survival, because a variant gene that protects against alcohol became almost universal against southern Chinese and spread through the rest of China in the wake of rice cultivation. The variant gene rapidly degrades alcohol to a chemical that's not intoxicating but makes people flush, leaving many people of Asian descent a legacy of turning red in the face when they drink alcohol. Now, I will admit that while I understand some of you find it entertaining, I was born and raised in Russia, in which I'm very, very, very sad to say the life expectancy of a man is 56 years. This is what a national epidemic of smoking and drinking will do. So, frankly, I'm slightly jealous of the fact that there are large groups of humans who simply can't drink in heavy doses, because the societal ill of alcohol overconsumption is tragically all too conspicuous in my mother country and many other parts of the world as well. I don't know how many of you saw this recent article in the New York Times about these beer stores that sit right next to Native American reservations and sell beer to them illegally. The spread of this new gene, described in January by Bing Su of the Chinese Academy, is one instance of recent human evolution, and in particular of a specific population's changing genetically in response to local conditions. I need to, now that I've given you a 2-minute sort of layperson summary, let me teach you this at the level in which UC Berkeley undergraduates should be able to know this. First, here's the reference. I encourage you to read it. Now, most of you won't be able to understand the results section, but you will be able to understand the introduction and the discussion, and certainly the abstract. The ADH1B arch, his polymorphism, and expansion of rice domestication in history. Well, first, you know, this is both tragic and funny. This is, of course, his Animal House, and Belushi's character goes, "My advice is to start drinking heavily." Otter goes, "Better listen to him, Flounder. He's pre-med." Ethanol, of course, must be broken down to be useful. This will not be on the midterm, but this, if you do take up our division biochemistry, this will. An enzyme called alcohol dehydrogenase makes acetaldehyde. Acetaldehyde is not pleasant. It must be immediately broken down by pyruvate decarboxylase to make pyruvate. Pyruvate, in some sense, is at the centerpiece of human metabolism. It allows human beings to burn all forms of sugar. Critically, in humans, the activity of this enzyme and this enzyme are coordinated. So there's a little bit of a pipeline or an assembly line. Alcohol dehydrogenase breaks down ethanol into acetaldehyde at about the same rate at which pyruvate decarboxylase makes pyruvate. This prevents the buildup of this toxic intermediate. In most people, well, actually, yeah, in most people, with a significant exception of people from large chunks of mainland China. This has a lot of text. I will just read to you the salient bits. The earliest agricultural use of rice was 10,000 years ago. It has been used as material to produce fermented food since the early Neolithic time. However, alcohol can lead to addiction, no kidding, and cause damages to human bodies, no kidding. Therefore, genes involved in the ethanol metabolic pathway might become the targets of selection when ethanol-containing foods and beverages began to be routinely consumed by Neolithic populations in southern China. ADH, the major enzyme that catalyzes alcohol, exists in two alleles, quote, wild type, and this specific polymorphism, which causes a change of arginine to histidine at position 47. So it's an allele of the gene. What does that change do? It makes the enzyme faster, much faster. So the moment you consume some alcohol, that enzyme starts to produce this much faster that it can be broken down into that. So the scientists hypothesized that the current regional distribution of that alcohol protective allele must be somehow correlated to where rice was domesticated. And here, on a map of modern-day China, are two sets of data. The frequency of the alcohol protective allele. This is 100% of the individuals cannot consume much alcohol. This is most of the people can consume much alcohol. You can see that this beautiful hotspot. And can you see the radiation in the frequency? So the further you step away from that spot, the lower the frequency of this alcoholism protective allele. And then they looked at the archaeological data to ask, where was rice first domesticated? Guess where? Right there. In fact, they were able to trace the timeline of rice domestication expansion from that hotspot in a way that makes it astonishingly coincident. Nobody alive was there 10,000 years ago when the ancient Chinese domesticated rice. But this correlation is just too ridiculously tempting to not then draw the conclusion from. Before I let you go, I'm going to read to you the meta-summary of what I would like you to know for the midterm from all of this. Lactose metabolism. We don't consume milk as adults, but humans do. And this, of course, is a consequence of dairy farming. There's clear evidence that the gene encoding lactase has evolved in the past 5,000 years under selective pressure to remain active into adulthood, and the genetic difference in one gene encoding a specific enzyme explains a common difference between humans. Ethanol consumption, alcohol, specifically ethanol, is toxic in high doses and must be broken down. There's a clear difference in the prevalence of an allele of alcohol dehydrogenase between human populations. The highest prevalence of the ultra-fast allele is in the part of mainland China where rice was thought to have been domesticated. Here's another genetic difference in one gene that explains a common difference between humans. I have office hours in 18010 right now, and I will see you Monday.